a protostome or whether you're a deuterostome. And here you can see that the blastopore becomes a mouth. So in animals like mollusks and arthropods and annelids, that first opening becomes the mouth. So proto, meaning first, and stome, like, stomata, like in plants, means opening or mouth too, right? So that gives you an idea of this one particular feature. First opening becomes the mouth. But not so with the others, right? With the deuterostome animals, that first opening, the blastopore, doesn't become the mouth. It becomes the other end of the digestive tract, the anus. And the mouth forms secondarily. So that's why it's called deuterostome. Because deutero is the root for what? For like Latin or Greek, I forget which one. It's like second, right? And stome is opening. So literally second opening. Okay? So very different features then are derived from that early embryological event of first opening in the hollow ball. So that's what number one is all about. Number two is even more basic, actually. And it occurs much earlier in development. So, you can kind of think of this as being like 2-cell, 4-cell, 8-cell stage in the developing embryo that you see in this table. And they look pretty similar, these two different types of division or cleavage patterns, right? Until you get to somewhere between the 4 and the 8. So somewhere's the processes between when it's a 4-cell state and an 8-cell state, something differs, doesn't it? In terms of how they divide and how these cells cleave. So that you can pretty much easily tell that spiral cleavage, this pattern by protostomes, has a different outcome than radial cleavage, as you can see here. What are some things that are different about now the 8 cells here versus the 8 cells here?
Bilateral.
Okay, good. Could you say that again?
They're bilateral.
Bilateral?
Symmetry.
Oh, one of them has more symmetry like than the other, like this one you're saying?
Mm-hmm.
Uh-huh. That's a good point here, is that in this 8-cell stage, right, it seems as if all the 8 are all kind of lined up, right? So the centers of the 4 that were just added line up with the centers of the 4 below it, right? That's what you mean by like symmetry? Mm-hmm. Whereas, is that the case for the 8-cell state here? No, right? It's the centers of these 4 new ones almost line up in the furrows, the places in between the cells of the 4 older or original ones here. Can you guys see that? So it's almost as if someone like took these and then slightly twisted them as if they're on a spiral to get into this position, right? So that's, of course, one obvious difference. What's another obvious difference between these 8 and these 8 here? The size of the next 4 here are, of course, much smaller than the 4 below it, whereas these are more similar in size. So we've then just described some of the basic differences in how the cell size and cell location, depending on whether you cleave in a spiral pattern, whether your cells cleave in more of a radial pattern. And so that gives rise to then sort of different relative positions of different cells in the body. Okay, so that's spiral versus radial cleavage. So again, protostomes, things like clams, things like polychaete worms and spiders have this type of cleavage, whereas sea stars and chordates have this type. And then these other two describe how the actual phloem formation in the blastula stage is quite different. So even having that body cavity that is, of course, the characteristic feature of coelomates, the way you get that is different over the course of animal development. And basically it shows you that one way you get by splitting of some early mesoderm, and the other one is you get it by outpocketing of early mesoderm, these little ear-like structures here. But I'll leave you to sort of look at the details that are in your book. And then there's some differences down here in number 4 in the determination of cell fates. That is, when does a cell in a developing embryo receive the information to differentiate into a muscle cell, a heart cell, a brain cell, a nerve cell? When does that happen as the animal is developing? And that's different depending on whether you're a protostome or whether you're a deuterostome. Okay? So, again, I don't expect you guys to know 3 and 4 in any terrible, really detailed way, but just in a general way. I'd like you to know 3 and 4. And of course, 1 and 2 are we talked about it being pretty fairly easy to recognize. Okay? So, all these features in embryological development then are wholly different, even though we're all coelomates, right? All those animals that we talked about as being coelomates share that common body plan, but the way they got there, their path of development could have been wildly different, as you can see here. Okay. So moving on then to our model, our representative of a deuterostome phylum is of course the Echinodermata. And you've seen that in lab already, and here's some representatives here. What I'd like to point out though is that the echinoderms, although they're related to this other deuterostome phyla, the Chordata, it turns out they're more closely related to a really small group of deuterostomes called the Hemichordata. So that's the phylum. Hemichordata. And they're weird-looking animals. Sometimes their common names are listed. They look like kind of wormy kinds of things, acorn worms or terebranchs. They're burrowers and substrates and sand and soil. Don't really look anything like what we truly think of chordates. And they're actually the closest phylum, the closest kin to the Echinodermata. The reason why chordates are not the closest has to do with the larval morphology or the larval condition of both the hemichordates and the Echinodermata. So what you see here are 2 different larvae. This one, do you remember what this one is? The Bipinaria larvae? What will that eventually develop into? Sea stars, right? The class Asteroidea. So this is an example of, say, early sea star larva here. And again, just like I've told you before, I'm not expecting you to memorize all these different parts, different stomach and ciliated tract. That's not my point. in putting this up. I want you to then take a look at this other type of larvae over here called a Tornaria larval stage. They look pretty similar, don't they? They have kind of similar features. They have these sort of tracks, and so based on the similarity of their larval forms, the larval of the hemichordata here, right? Larval echinoderm here, that is the sea star. That's why they're thought to be such evolutionary ties between these two groups as being sister phyla to each other. Because they have very similar and kind of as sort of a foreshadowing, shall I say, with some of the information that you will actually start to study when you get to the second half of the course. We'll be concerned with looking at a lot of the developing larval features when you get to looking at the early chordates and the protochordates. So when you get to this group, the chordates and the duderostomes, a lot of the larval features play a big role in terms of both not developmental significance, but evolutionary significance too.